Okay, so as we look at this digestion that takes place in these sponges, I had mentioned maybe grab a book, and I see a lot of you have done that. We'll be utilizing that here in just a moment. And it's more so a visual aid because I think a lot of the content that we really need, we placed up on the board. We just need to, let's say, put it into context. So where is it that in each and every one of these cells that when we're talking about phagocytized, that's at the very bottom there. So algae is moving through this sponge because of the water currents, and the choanocytes, it's not that they regulate them, it's because the flagella are attached to these choanocytes. But where does this digestion slash phagocytation, and where would that take place in the sponge? It's in each and every one of the cells. Choanocytes is not specific enough. Okay, that's too far down the line. In the colar, okay? Because what you see happening is, well, you can't actually see it, but what we had talked about yesterday, okay, is that around this circle, we use the example of a nickel, okay, and that now we're putting these choanocytes in here, and we could go all the way around. Then I think we drew rectangles like this. I'm not going to put them on all of them, or else just going to run out of space. But now we have these flagellated chambers, you could call it that because of these flagella. So the water and algae is moving through here. The algae gets caught by these flagella that are just moving because of the water current, okay? And I believe it says something along the lines they get trapped by mucus. That's not the first time, or excuse me, that's not the last time we're going to be talking about that, because that also happens in something very similar with, we could say, that of mollusks like slugs and snails. I know you had mentioned that before. And also that of clams, but that's a little later down the line. So this algae gets trapped on this flagella, and it moves down into this structure. And you correctly identified it, calling it the what? The colar. Okay? So I'm just going to put CO for colar here. And then how would that be different than what we see here? We just verbally said it. These are your choanocytes then. We put CH here all the way around. Okay? So as this algae gets moved down, because it's trapped on this mucus, down to the colar portion, that's where this digestion takes place. Now, that occurs in each and every one of those cells. Is that what we would consider an advanced type of digestion? No. And why would that be? Well, yeah, because they don't have organs. They don't have a pumping mechanism, a bloodstream, arteries and vessels to actually move some of this content from one part of the animal's body to the other. Because very simple animals don't move. They're filter feeders. That's two characteristics. How about two more that all sponges have in common? They're sessile, and they got pores. So those are your four characteristics. That there's many more that we could use, but those would be probably the four that would stand out. Okay? So the biggest hole in a sponge, we're just talking about their porous. Which one's the biggest hole? Yeah, the osculum that will be located up on the top. Okay. Okay. So we're going to add to your drawing again, because when we talk about support, we need to put that in there, because even though they don't move, they still need to be able to, let's say, withstand different types of water currents. So again, don't let this be misleading. The author says it's pumped through here. Typically, when we think of pump, there's something physically pushing that along. Okay, that's not the case. It's just the ability of, you could say, the choanocytes or the flagella on the choanocytes being able to move to regulate that water. That might be a better way. Feed primarily on particles suspended, the water, and regulated through the canal system, or again, it's not a pumping mechanism that we would visualize. Now, I could certainly understand sitting in here thinking these are not very advanced animals, and I would agree. Maybe it's not that interesting to think about, and I would agree. But because of how they feed, what might happen in certain parts of the, you could say, seas where these are located? Because of how they feed, what would happen if you remove sponges from the water? Well, that's what would happen to the animal. I mean, what might happen to the ecosystem itself, do you think? I didn't think anybody would know that or think that. What made you think that? Yeah, because of the algae that they would eat. That is correct. Now, there's actually a type of symbiotic relationship between sponges and other animals. What might that be? In other words, one organism is benefiting from another, okay? So what that might mean is, okay, you're throwing your key blocks where you want to or need be. Man, that running back really, or quarterback sneak, they're really looking good. Well, that's because the offensive line is opening up a big gashing hole for them. So whoever's carrying the ball benefits from that. And then hopefully it's then reciprocated, that the idea that you benefit from the victory, you could say, or something to that effect. Just knowing that you're doing your job, you benefit from that. And anyway, for animals that are a little more advanced in nature, we talked about this yesterday. What is it that would hold mammals up? Skeleton. It's going to be their skeletal system. Now, how many of you are, I suppose we could say, deer hunters of some sort? Okay? You actually go and do that. Are you successful every year? Is it frustrating? Yeah. Does someone say, well, look what I got? Yeah. I can see how that would be irritating. Now, is it that they really want to show you what they have, or is it where, ha, 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 this is what I got and you don't? Maybe it's a little bit of both. Yeah. Maybe it depends who it's from. Okay. So what is after you bring your catch home, I don't want to say catch, but your limit, I suppose, how is it that you actually drain the blood out of these animals? Yeah, you would hang them up by their, back feet? Now, you just don't put them up on a hook and put it inside their hind limbs, or I suppose you could, but then you're going to do a lot of damage to the animal. But I would be led to believe there's some really tough connective tissue back there. And it's probably the same name that we have for our connective tissue above our ankle and foot. Yeah, the tendon of Achilles, or the Achilles tendon. I would think that's what you might call it in the deer as well. So it could be something, an animal is fairly heavy, but you're not going to rip through that tendon, are you? Because it's, again, a really rigid structure that has lots of protein in it. And It's not that it provides support for the animal. It kind of does because it's attached to muscles. But in this case, different forms of collagen, as you can see here, is a protein that would provide support for these animals, because you got to remember there's can be rough seas out there, and just because you're below the water surface doesn't make you immune from rough seas. It can be just as rough down there as it can be up at the surface. So as you're having these water currents push through these animals, you have to have a really good support system to hold them in place. And that's what collagen does for these animals. And not only is it collagen, I think another term we call is spicules. I would think that's got to be coming up here soon, or maybe we've had it already. If not, I would be led to believe that is a vocabulary term for you. I'll let you finish writing that first. So you gentlemen, will you approach Friday night's game any different than any other one just because it's a bigger field, or does that not matter to you?
I think it's a little easier.
I think you said that earlier for what, your passing game, or okay. But then also going to make it a little more difficult to defend, and that's the exact same point that you just made. Would you want to practice on that field if you could, or does it not make any difference to you?
We got 100 grit. We got cut it longer.
Oh.
There, so we're practicing. But yeah, it makes it.
Okay. All right. So then, just a moment here. All right. So, one of your vocab terms, what might be structures that make up the skeleton? Yes, there's was it spongin that we had back here? Spongin is one of them, and what might another term be that makes up the skeletal framework as you're looking at your vocab terms? Collagen is going to be what's going to make up that. Yes, that's true, but it's not the actual structure. In other words, collagen is found in ligaments and tendons, but it's the collagen that doesn't hold the body up. It just holds it together. Should say something along the lines of structures that make up the skeletal framework. I think that's what it should say. Maybe it's not on here. Oh, I guess it is not. I'm just going to do a quick. Oh, because it's a related term. So when you look at spongin, okay, that would be the material that makes up the spicules. That's what we're looking for. I was thinking that was somewhere along the lines of maybe a little earlier. Spongin. Okay. So spicules makes up the skeleton or framework. Okay. I thought that might say it in here. And then the spongin that makes up the spicules. So it's kind of like the type of collagen and spongin is a protein then that makes up this structure. Okay? Or helps make up the spicules to hold the sponge together. Okay, so we're past this. We'll wrap this up. I think this is where we were, right? Yeah. Okay. So now, what does this mean to you? Now we can include everybody because everyone has had this content in biology. What about diffusion? It is something to do with cells, that's right. Can we be a little more specific though? Okay, maybe combine what you two had said. I didn't quite hear it.
The cells are splitting apart.
No, that's not dividing.
I said that it works by working up or down the size gradient.
You're right, you just need to pick one.
Okay.
Was that a guess?
Yeah.
Was it a good one?
No.
Yes. Yeah. So when materials move down a concentration gradient, okay, that's how materials get into and out of either whether it's our cells or even peripherin cells, okay? So when we compare diffusion to that of water, which is osmosis, to movement of proteins and sugars, which is facilitated diffusion, there's no need for active transport, because active transport is a means that makes muscle cells work. Because muscle cells will not work unless they are stimulated by something. And that is the nervous system, right? Back corner? Sure, and it's called what we call wave potentials, okay? But none of that is present in here because these animals don't move. So the only type of movement they need is just materials in and out of there. So what might be diffusing into and out of those cells? If it's not sugars and proteins, they still need this type of substance to do cellular respiration and give themselves energy, which is oxygen. Okay? So the reason that would make sense is because we're talking about respiration here. Diffusion is really small molecules like blood gases, okay? Although in this case, again, there's no blood, but there are gases. The oxygen is extracted from the water, then the carbon dioxide is going down that concentration gradient and then being released. Okay, so I think that is the last one here. When we think of these simple animals, we really believe that as far as reproduction is concerned, they might or must be clones of one another. Well, I would believe that they can, okay? But this here is a rudimentary, or you could say an older version of what sexual reproduction is, because you have both types of sex cells present. Because when we're talking about asexual reproduction, that is more so associated with that of bacteria. Very, very simple animals. Well, not animals. Very simple organisms. That's what I meant to say. Okay, so we may put some additions on this week because when we say types of sponges, that was just a very vague term of saying we're talking about the different types, which to your knowledge, what are the three types? Now, you wouldn't know that. You would have to look down and see once what they are because it is in your handout. Okay, that's being too specific. I mean, you're not wrong, but okay, that's the phylum. That's one. Asconoids is one. Sycanoids is two. And leuconoids is three. So what we've been doing is when we're drawing these, I want you to look at page 115. So in our purposes, okay, what type of sponge do you think we've been drawing because of the different types of colar cells, spongoceles? Do you think it is it the top one here? Is it one like this, or do you think it's one like this? If you look up at the board, that would be a clue. If we look up at the board, that might be it. Where do you see something similar to this? Second one on page 115. So you say asconoids? Okay. Where, or I should say, excuse me, maybe a better way of looking at that. I mean, they do have this in common, and yes, it does have a spongiocele, but does this sponge have anything in there like this? No, it does not. So it's not asconoids. Say that again. It's leuconoids. Okay? So when you look at the leuconoid down here, okay, notice it's got these chambers that go in a circle like this, very similar to what we see here. Okay? So we wanted to give you that visual representation. Now, the asconoids and sycanoids, yes, they still have these structures, but I can understand why you would say sycanoids. It's just that that's not quite in a circle like what we've done here. It's the leuconoids that have all these different types of chambers inside of them. Okay? So, moving forward then, when we talk about the phylum Porifera, because this is what we're going to continually add upon throughout the semester, and this is where it begins, because some of you may remember this from 1 and 2 years ago. Of course, when we have the kingdom, okay, whether it's from biology or this class, what kingdom are we talking about? This entire class is about what? Well, okay, we're giving answers, but I mean this entire year will be about what? Someone said it, it's about animals, so Animalia. Now, from last year and 2 years ago, then, now this part's wrong, okay? We would have put this because chordata means backbone. Do sponges have that? No, they're invertebrates. This means vertebrate. Then we would have went to here. Okay, so we got a kingdom, we got a phylum, now we got a class. Then that's where you went to your different orders, whether it was a marsupial, a monotreme, okay, a proboscidean. Okay, then we also had, let's see here, rodents. Then we had, I was going to say gophers, but gophers are rodents. Then you would have artrodactylians. Those are, okay, even-hooved mammals. Then here are your odd-hooved mammals, Paracedactylia. So each and every one of these is a different order of the class Mammalia within the phylum Chordata. Okay? But in this case, all of this is going to be erased except for this, because we're still talking about the animal kingdom. And now we can say, what phylum are we talking about? Okay, that's too vague because there's lots of invertebrates. Yeah, this phylum we're talking about this chapter is Porifera. Okay, so then these poriferans, okay, we could say get broke down to, this is the backside of your vocabulary. Okay, oops. Your vocabulary looks like this, of course. Then the backside, you have this handout of this handout. Yeah. So we have a class, Calcera, then. Okay. And then these we want to associate as being very simple. So we would call these are asconoids. So I don't know what, I would be maybe just writing this on that sheet for sure, or if it's in your notebook, that's fine too. Okay. And then, let's see here. I don't want to run us totally out of time either. Okay, so we're still in the same phylum. Okay, and then I guess we would have to extend this down because we want to stay in the same class. Okay, which is, what is in order of this? What's the order for Calcera? Okay, Homosolia, I believe. So we're just going to put order. Okay. And then, is that the only order for this class? No, there's not. So we still have Calcera, but the other order is, again, what? So then we put order Heterocilia. Okay? So we just want to start breaking these down because we're going to associate this order more so the cycanoids. Okay? So the asconoids and the cycanoids belong in this class Calcera, but then they are in different orders. We'll move on to that of the leuconoids. We still have some more characteristics that we want to put down in here and the animal, but this gives you somewhat of an idea that will continually build upon this content, because this is what your final is over in December. Okay? These characteristics, and there is a lot of them. Okay. So I will probably have short answer papers for you tomorrow. I don't think we'll have a vocabulary evaluation this week. We'll save that for next week. And then if we assign that on Thursday the 12th, maybe by the 17th, we'll have a vocabulary evaluation, and then maybe review on the 18th and have an exam next week over the sponges. All right, that's all I have. We'll catch up to you next time.